The cell membrane is also known as the plasma membrane and is made up of bilayers of phospholipids, glycolipids, cholesterol, and proteins. The biological functions of the cell membrane are to provide support and to act as a barrier for the cell. Cell membranes are the semi-permeable boundary between intracellular and extracellular fluid. Basically, cell membranes are what keeps things out of the cell out, and it keeps what's inside of the cell inside. During this video, the educational goals are to identify and define what a phospholipid bilayer is, understand how phospholipids arrange themselves in the bilayer, and understand the fluid mosaic model. First, we will address what makes up a phospholipid bilayer. Phospholipids consist of a polar head and two fatty acid tails. To get a good understanding of the components of the phospholipid bilayer, let's break the words down. Phospho, which means that there is a molecule containing phosphate, and lipid, which is going to be addressed in this video as a fatty acid. And of course, there is a bilayer. Now let's break each of these components down, starting with the phosphate. Phosphate, or PO4 3-, is going to be present in phospholipids. These phosphates are attached to an alcohol and the glycerol portion of the phospholipid molecule. These are what make up the polar head region because they contain many polar covalent bonds, especially within their CO and HO bonds, which we'll see later in the video. A quick review of polarity shows us that these polar heads of the phospholipid are hydrophilic, water-loving molecules. Think P for phosphate head, P for polar, and P for philic, which is the Greek suffix for affinity or love. Keep this in mind throughout the video. Next, let's look at the lipid portion of the phospholipid. Lipids in general are biomolecules that are water insoluble or hydrophobic. Phobic, which is the Greek suffix for fear. Earlier I said we'd be addressing the lipids as fatty acids. So, fatty acids are the carboxylic acids that contain an even number of carbon atoms, usually between 12 and 20, and are built from two carbon hydrocarbons. Nonpolar covalent CH bonds make these long hydrocarbon chains nonpolar overall and promote their hydrophobicity. CH2. These long fatty acid chains are represented as nonpolar hydrophobic tails to the phosphate attached to the alcohol, polar heads. On a side note, I want to briefly address the backbone that holds the polar head to the two fatty acid tails. When a fatty acid tail loses a hydrogen atom, it becomes a carboxylate ion here, and it also becomes amphipathic, which means it now has hydrophobic here and hydrophilic counterparts. This carboxylate ion can then form the two classes of phospholipids. The carboxylate ion can join with the glycerol to form a glycerophospholipid, or the fatty acid carboxylate ion can join with the sphingosine to form a sphingolipid. To the right of each phospholipid class, you can see the chemical makeup of the structures. Notice the CO and HO bonds in the polar regions. These two classes of phospholipids are present in the phospholipid bilayer. However, during this video, I will not distinguish between the two. The bilayer implies that there are two layers of these amphipathic lipids present. So let's now take a look at how they arrange themselves. Since we know that like attracts like, then it's easy to understand why the polar hydrophobic phosphate heads will want to interact with one another. On the same note, these polar phosphate heads will want to arrange themselves so that they are closest to areas where there's water, right here and right here, present, such as the internal and external sides of the membrane. The fatty acid hydrophobic non-polar tails will also interact with each other, staying as far away from water as they can. Here is one way to think of this arrangement. Think of a class full of students. There they are, in a room that has a mirror as the entire floor. There's the mirror. Now, picture the students standing next to each other with enough room in between them so that they can spin around like a ballerina, that's what she's doing, and move around laterally, walking and standing straight up, like what they're doing, walking to the back, walking to the side. If you could see through one of the classroom walls and look at what's going on inside, you would see the students' legs, there they go, there's the mirror, both their actual legs and the reflection, arranging themselves together. The students' legs represent the hydrophobic nonpolar fatty acid tails that interact. The students' heads represent the phosphate heads of the phospholipid bilayer that are attracted to the extracellular here and intracellular here fluid. The reflection of the students' heads illustrates how the phosphate heads arrange themselves towards the intracellular portion of the cell and extracellular portion of the cell. Remember that there was enough space for movement in between the students, or phospholipids. That space and the continuous movement of the phospholipids and inter- and intracellular proteins, glycolipids, and cholesterol is what makes the cell membrane dynamic and continuously changing. The dynamic nature of the cell membrane is attributed to the other main components commonly found within and along the phospholipid bilayer. These include cholesterol, hydrophobic molecules used to synthesize hormones and add rigidity to the membranes, here, glycolipids and glycoproteins, with lipid sugar and lipid protein complexes that act as identification tags for cells, and other various proteins that help the cell transport nutrients and wastes into and out of the cell. Here you can see integral proteins. The constant motion of these molecules coupled with the dynamic motion of the phospholipid bilayer itself represent the fluid mosaic model, where phospholipids, glycolipids, proteins, and cholesterol are all structural features of the mosaic-like cell membrane. I hope this video has given you a better understanding of the basic structure of the cell membrane's phospholipid bilayer and a deeper meaning to this song.